I'll take that. Hello. Hello, Dr. Pradnya, can you hear me?
Hello.
Yeah.
Is that Dr. Mahendra? Yeah, we can hear you.
Yeah. Thank you for joining Dr. Pradnya and Mr. Nandakumar. Thank you. We will start in a few minutes from now.
Yes, sir.
Yeah.
Hello. I'll say something and then I'll hand over to you.
Okay, sure.
So I'll just start the event and invite you for a special announcement. Okay, so let's just settle in. Maybe there's lots of seats available in the front if you guys can come in. I am hoping the auditorium today will get full. Okay, so I welcome one and all for this edition of the Wednesday Colloquium. And we have our speaker today, Professor Anusha Shankar, who has come all the way from Hyderabad, TIFR Hyderabad. So, but before we formally start the colloquium, we have a special announcement from the department chairs. So please settle down and we'll hear it from them.
So the special Wednesday colloquium is part of the DBS annual talks, which is a annual event that we have where we collect to ruminate and reflect and think about what we've done over the last year and also discuss exciting avenues that we are going to follow in the year that comes ahead. So Mahendra and I are actually super thrilled that we get this opportunity to announce an endowment to the Department of Biological Sciences made by two wonderful people, Dr. Pradna Sarvade and Mr. Nandkumar Sarvade, who are joining us from Canada. And hopefully they are there online. We would have loved to have them present here personally, but we're really happy to have them join us online from Canada. This endowment, which is a special endowment to DBS, comes with a very specific focus. It is in the name of Sri Jnandev Govind Bhosle, Dr. Pradnya Sarvade's father. I'll tell you a little bit about him in a minute. But before I do that, I'd like to tell you what this fellowship and endowment is for. It's divided into two components, and maybe I'll just ask Mahendra to describe those two while I then person it's named after.
Right. So there are two components that we are going to use this fellowship for. One is to give 3 to 4 travel awards for students who want to visit abroad or within the country for conferences. And more importantly, one fellowship to a student who are actually on extension, who has finished his or her fellowship, but is working on a challenging project, defending comments of an important paper or some landmark paper. So such students will be supported via this particular fellowship.
So we're very thankful to Dr. Pradnya Sarvade for initiating this endowment and fellowship in the name of her late father, Sri Jnandev Govind Bhosle. Sri D.G. Bhosle, for whom these fellowships are named, was a self-made man who grew up in a farming family based in Khamgaon in the village of the Sholapur district of Maharashtra. He grew up in pre-independence India and was one of those people of that generation who placed great emphasis on education and the importance of education to planning one's life trajectory. He grew up in and came to Mumbai, where he came in contact with many rising and social political leaders of that time. And he was also associated with independent journalism and worked as a journalist with Navkal, a Marathi language newspaper of that time. He then transited after his time as a journalist into the teaching profession, and that is perhaps why his lifelong commitment to education comes from. He taught in schools in Mumbai, later in Thane, and then in Lonavala. He emphasized teaching, lifelong emphasis on teaching. And he lost his wife very early in a railway accident, was a single parent who managed to raise a wonderful young woman at the same time as absolutely staying committed to his career in education. He was also a great devotee of the Marathi saint Sant Jnaneshwar. In his name, Dr. Pradnya Sarvade, who's actually a medical doctor and then transitioned from that to the police services, and has been visiting DBS over the years and has visited and spent time in Dr. Krishanu Ray's lab in DBS. She's the one who's instituted this endowment and this fellowship. So this is the first year. We're very happy to have Jairam actually here with us. And if you can just say a few words to Pragyan.
No, not at all. Yeah, so I'm very glad you're here with us, virtually at least, if not in person. And, as I've said before, we are, as an institution, very grateful for this gesture. And we think it will make a major impact, on the students who will benefit from it. And so, thank you both again.
So we have a committee that looked at potential applicants for this year. The travel fellowships will be announced a little later because people will be applying for them across this year. But we have a candidate this year. We'd like to use this opportunity to announce the candidate. So Kamaldeep Singh is going to be the first recipient of the Sri DG Bhosle Fellowship. So Kamal, if you can stand up for a second so that we can give you a round of applause. Kamal has been working on an interesting project looking at the impact of insulin on transport in using the fly as a Drosophila as a model system. So thanks, Kamal. Yeah. Okay, so we can hand over now. Thank you, Pratnya and Nankumar, very much, and look forward to seeing you. Yeah, would you like to say something? You're on mute.
Thank you, everybody there. Thank you, Dr. Vidita, Dr. Mahindra, Dr. Jairam, and all the faculty of DBS and TIFR. It is I'm extremely glad that we have the first recipient of the fellowship, which I instituted in my father's name. And Kamal is one of the is an excellent recipient for this first award. I'm really glad that he has got this fellowship. The whole idea of giving this money was not that I just had money to give around, but TIFR, I felt, is one of those institutions which really make the nation proud. And whatever support in terms of acknowledgement of its work through these kind of endowments, grants, which I could contribute to, it has been my pleasure to do it. My father, as Dr. Vidita mentioned in the little introduction, he really pushed for science and mathematical education and thinking, which was, I think, quite extraordinary, being from that kind of a period where not many, at least in rural areas, people did not put children in schools. But he came from that background and he pushed for that kind of an idea. And this grant is nothing more than some kind of an acknowledgement of that concept and idea. And I hope the greatest of scientists that we have in this country come from TIFR, and they will take such an idea forward, not only progress in science, but also lead that progress in science for the benefit of society and give back to society whatever society gives into making of them. Thank you very much.
Okay. Yeah, I thank you again. I think this is quite a fantastic start to the colloquium proceedings today. So thank you very much for doing this at the start of the colloquium. And we have a fantastic packed house for you, Anusha. So before we start, I would like to invite a dear friend, Professor Ullas Koltur, to introduce Anusha.
Yeah, I'll already say that you're going to be in for a feast of biology. Take it for granted from me. So I used to know this almost by heart not so long ago, but maybe I'll still read it out. Okay, so I'll quickly tell about where Anusha comes from, and then maybe describe a little bit about why what she's doing is so exciting in general for biology itself, but also how it has relevance in the current day and time. Anusha is an ecologist and does a lot of physiology, and that's a very unique combination. I'll say a few words about that. She did a PhD from Stony Brook, essentially looking at doing field physiology using hummingbirds, which is a very interesting species of birds. I think many of us would have probably seen them in National Geographic or other some of these documentaries. Subsequently, she went on to do two postdocs. One is in Alaska, the other one is Cornell, continuing her interest in looking at physiology in ecological niches. And for postdocs, she actually chose very two diverse, distinct questions. One was more of affective disorders, which are more behavioral in diurnal Nile grass rats. The other one is going more molecular, again, back into the hummingbirds, looking at what dictates the physiological responses at a molecular level. Now, why is this particular field interesting? I mean, there are several ecologists that we have. Of course, that number could have been much better in the country, but we do have some fantastic ecologists in the country. But ecological physiology is a very niche area, and not many people, as I understand, work in this. This is important because as much as you want to understand about how ecological nature shapes organisms, there is very little understanding of how physiological adaptations happen, particularly given that there is a lot of human activity and a lot of change in the environment that is taking place. So that's why this particular field of work is relevant for now and in the future. The ecosystems are rapidly changing. We understand very little about how organisms adapt to these. With a specific question that I think she is going to deal with in the initial years is that looking at metabolic adaptations and energetic adaptations, because these are ultimately the ones that is important for all of us, but become even more relevant in the context of organisms living in the wild, because access to resources in the form of nutrition and their energetic demands are not very clear. I mean, talk about whether it is long distance migrations or changing temperatures and whatnot, or seasonal variations. So in that context, what Anusha does is very unique in the country and probably also in the world. There are very few people who work in this. And I think she's going to talk a little bit about that. And it's really my pleasure to introduce Anusha here. Thank you.
Thank you. Getting a spam call? Okay. It's really an honor to be here. Whoa, that is a packed room. Wow. It's a lot of you. It's an honor to be here. I don't think I would have ever imagined being at TIFR as faculty, and it's really strange that I am that now. So I joined TIFR Hyderabad about 11 months ago, and I'm really enjoying being in a department for the first time that's not an ecology department. That's a broader biology faculty, because it leads to all these opportunities for interactions and collaborations and talking about science from a very different perspective than I've been used to all these years. So I'm really enjoying this, and I'm looking forward to talking to you all who are not just biologists, but also presumably physicists and chemists as well. Okay, let me see if this works. Okay, so the title of my talk is Budgeting Heat and Energy, How Do Birds Do It? And I call myself a physiological ecologist, but I've been working towards becoming a broader, more integrative biologist and using a variety of methods to answer a very fundamental question of how animals adapt to their environment. So some of my history, the further back than what Ullas mentioned, my growing up kind of history that led me to these questions is just watching a lot of animals in the wild. So I did some early field experiences during my bachelor's degree in Assam, where I volunteered to study these hulock gibbons in the wild. So these are gibbons that swing through the trees. They mate in these monogamous pairs. And there was this one female that had been orphaned in the wild because her parents had been poached. And so this rehabilitation center was taking care of her until she was about 4 years old, and they wanted to rehabilitate her by releasing her into the wild. So I was studying her behavior before and after her release. And then I did a project where I tracked king cobras in Agumbe in Karnataka. And I started just looking with my little point and shoot camera at all the little things on the ground. I didn't look up like a bird person is supposed to do. I just looked down because that's what my camera lens could capture. And I paid a lot of attention to the butterflies and the ants and the spiders and the little insects all over the place. And then I ended up doing a master's in Pondicherry University and studying these birds called hornbills. You have hornbills here, right? You've seen these? So these are Malabar pied hornbills. And they're very ecologically convergent with these toucans, which are in South America. The hornbills in India nest in tree cavities. And so they do this really strange thing where the female goes inside the cavity of a tree and seals herself in with mud and feces until there's an opening only about this big for her beak to come through. And so the only source of food she has for 1.5 to 3 months, depending on the species, is the male coming and bringing her food. And so she lays eggs, hatches her chicks, and rears them completely inside this tree cavity and undergoes a full molt of all her feathers.
It's a very extreme life strategy, and they depend on these large trees to do all of this and reproduce. And those large trees are getting cut down. So I was trying to study nest site selection in a very fragmented habitat in the Ratnagiri district in Maharashtra. And I took a lot of pictures of little bugs while I was doing that as well. So what strikes me when I look across these organisms that I've studied is that there's so many different ways to exist in this world, right? There's so many ways to exist and to deal with environmental change and to adapt to our environments. And we all do it in such different ways. And I've lived in so many of these environments. I've lived in Alaska where it was -40. I've lived I'm from Chennai, which is very different. And so we have different strategies. We've created our environments around us, but these animals in many ways have to do that with their own bodies. They can't really modify the environment as much as we do usually. So I want to study how animals, what mechanisms animals use to adapt and deal with environmental change and with variable environments. And I think this plays into a lot of what Shraddha was talking about, the traits. I think she had a very similar starting to her talk, right? There's so many traits that allow the animals to exist in their environment. And also it'll touch a little bit on sleep and what Nitin talked about in the morning. So this is the broad framework, just to orient you before I go into some storytelling. I am interested especially in heterothermic animals. So you might have heard of endotherms like us. We generate our own body heat. We maintain a high body temperature. There's ectotherms like lizards that allow the outside air to decide their body's temperature. And I'm interested in these animals that can do both. They can move between endothermy and ectothermy. And I would like to do this in a comparative phylogenetic context, comparing across groups of animals that can do this. In my second postdoc, I started doing RNA sequencing to try to understand the genetic mechanisms that can underlie this phenomenon that I see at a whole organism level. And I want to do this in a free-living animal context. So it's very standard in biology to take an animal, put it in a lab, control everything, and study it. But I don't want to do that. I really want to study animals in their natural environments under natural temperature and light, because I think there's a completely different set of questions we can answer by doing that. And I'm really interested in how animals manage their energetic needs. So I'll put these all together piece by piece now. For the past 11 years, I've been studying these birds, hummingbirds. They're very small. How many of you have seen them? In person? How many of you have been outside India? Okay, the people who said yes to the first question and then no to the second question have not seen hummingbirds. If you've seen tiny little colorful birds flying around like this in India, they're sunbirds, they're not hummingbirds. So hummingbirds are only in North and South America. And you can see they're really tiny, they're extremely fast. You have to use some fancy slow motion camera to see their wing beats. But there's some 23 species at this one site in Ecuador where I filmed this. And so they're coexisting. They're feeding on nectar, which is just sugar water, most of the day. They also eat insects to get their energy. They have an extreme lifestyle because they're so small and they use up energy so fast that they're constantly within a few hours of death in terms of the energy stores that they have. So to give you a sense of this, I'll go back to, somehow I also have a Matt Damon. Where's Shraddha? I also have Matt Damon in my slides. Yeah. I don't know why. So Matt Damon weighs about 85 kilograms. And in case you didn't know that. And this graphic tells you how many packets of Lay's chips Matt Damon would have to eat if he had the metabolic rate of some other animal. So as himself, he probably needs to eat about 15 packets of Lay's chips to meet his caloric needs every day. We're just ignoring nutrients other than calories. Like, no, I don't care about vitamins and minerals and all of that stuff. So just calories, he can meet his daily energetic needs with about 15 packets of Lay's chips. If he had the metabolism of an elephant, he would need to eat about 4 packets of Lay's chips. So it's a very fundamental principle. As you get bigger and bigger, you need less energy per unit mass to stay alive, right? True for most of biology. If he had the metabolic rate of a hummingbird, can you guess how many packets of Lay's chips? 1,000? Just a little louder. 1,000? Anything else? 260? Good guesses, big range, 60 to 1,000. So it's about 600 packets on average. In some individuals, it can go to about 1,200. So you were very close. So this means that as a very small organism, they need a lot of energy per unit mass to meet their daily energetic needs, right? That's a good question. I don't know. It's a kind of beetle, I guess. Maybe it just like acts dead. I actually don't know. I made this many years ago and I don't remember. So hummingbirds somehow are so small, they barely store any fat, using up energy very fast, right? And still they've managed to live in a diversity of habitats. So they live in the cloud forests in the Andes. They live in the deserts in Arizona. They live in the high elevation Andes, 3,000 meters above sea level. And they live across, yeah.
Energy requirement in terms of base package against mass?
Yes, I will show you that. Yes. Yes. The question was, have I tried plotting lace packets against mass? So they've managed to diversify to all of these habitats, and there's over 370 species of hummingbirds that live in North and South America, mostly in South America. And they have a huge diversity of form and of function. So you can see this one with the giant bill. It's called the sword-billed hummingbird. It's the only bird with a beak longer than its body. There's also the crimson topaz, extremely colorful. The tiny booted rackettail is one of my favorites. It has little white boots on its feet. And the giant hummingbird is a whole 19 grams, and it fits in your palm. That's the biggest hummingbird there is. So the smallest hummingbird weighs about the same as an old 10 paisa coin. And this one weighs 9 10 paisa coins put together. So they're really, really small. And to get to your question, early on, I realized that the study of allometry was interesting for hummingbirds. And allometry, I don't know whether to say this Indian or American accented anymore, but it's the study of any trait, any biological trait, for example, with mass on the x-axis. We usually log both these axes. And early in the 1800s, people started studying it because they wanted to know if there's some universal laws governing all of life. Is there some universal scaling metric we can find that links bacteria to elephants and whales or something? And they started finding that most of these organisms, like for metabolism, this slope tended to be about 3/4 or 0.75 across all of life. And this is really interesting. I thought it was an abstract and useless concept as a field biologist, but it turns out it's very practically useful as well. There's, if you want, as a veterinarian, if you want to dose a horse and you know how much to dose a dog, you can try to use these relationships to predict what to dose other organisms, right? So then in the 1990s, people started finding that it would really depend on these scaling relationships really.
The graph has been shown with breaks. I had a couple of questions on the first graph. How significant those breaks are, and if they are significant, whether there was a understanding as to why at those mass scales there is, this offset.
Yeah. So I think depending on, there are many camps. It's a very controversial subject, allometry. I think the physicists who study it want one number and say that it's all because of fractal branching patterns in the veins in your body and things like that lead to a 3/4 law. But there have been people have been looking at it a lot more, especially in the 1990s and 2000s, and are finding that it really depends on what taxon you're looking at. And so you can go, if you can look within a species and really zoom in and find different patterns, which I'll talk a little bit about. But I think if you want to kind of blur your eyes a bit, the 3/4 law can apply across all of life. So the slope is the same, but the intercept is different for these different groups. I don't know if they found significant differences in that particular paper. Yeah, you're talking about one species though. This is a scale where that all doesn't matter. Like that one species business is not what they're looking at in this.
You just excluded it because
I didn't. I wouldn't do that.
Okay.
I'll show you what I did. That doesn't exclude all the outliers. But if you look across all of life and you have reptiles versus mammals, I mean, those outliers are maybe one point, right? We're looking at broad patterns and the best fit lines. And those, they'll have some error that might be more or less depending on the size of the group. But the overall pattern is what people are trying to understand in this global kind of analysis. There's like 11,000 species of birds. What's one godwit going to do for that pattern, right? So what do these allometric relationships mean? If you have energy on the y-axis and mass on the x-axis, a slope of 1 on a log-log scale tells you that a large animal on this line has the same energy expenditure per unit mass as a small animal. Are we good? So if you have a slope that's a little bit lower than 1, that means a large lizard has a spends slightly less energy per unit mass than a small lizard. Mammals have a lower slope, birds have an even lower slope. So big bird spending less energy per unit mass than a small bird. And hummingbirds are birds, so we'd expect that they have something around this slope, but this is where it starts to get more complicated. If you zoom into just one single taxon or one group, you can find different relationships. And I found this one really strange. So in the 1990s, this Kenneth Nagy published this paper with data from 6 hummingbird species, finding that the slope for their scaling was close to 1.21. And this is really strange because it means that a large hummingbird energetically doesn't make sense to exist, right? They are spending way more energy per unit mass than a small bird. So why would you be a large hummingbird? There is only one really large hummingbird, the giant hummingbird. But I wanted to know a little bit more about this relationship and why it exists. Does it hold true for more species if we include data for more species? But how do you do that? You can't put a Fitbit on a hummingbird. They're way too small. And imagine this watch on a thing that's like 10 paisa coin weight. It wouldn't work. So we have to find other ways to measure them. And one way is to catch them. So we use these feeder traps. They feed on nectar very conveniently. So we can put these traps around the feeder. And the birds look like this. You can see the iridescence on the throat, right? Changes color depending on the angle. I'll talk more about that later. And then one method we can use is called the doubly labeled water method. And I think it's a genius method. People are using it for nutrition studies like Ullas and Mahindra with Arumda, where you basically take a double isotope of water and you feed it or you inject it into the animal, let it equilibrate with the body's water, collect a pee sample or a blood sample, let the bird go. 24 hours later, you try to catch the same individual again. That's a whole separate story, but you try to catch the same individual again and collect another pee sample or blood sample and look at the change in these 2 isotopes. And because you lose oxygen through your breath and your pee, and you're losing hydrogen only through your pee, the difference in rates of loss of these isotopes tells you how much carbon dioxide they breathed out in the time that they've been away. And so you know in 24 hours how much carbon dioxide they breathed out, which is a really good proxy for how much energy they've spent in 24 hours. So you do this to get daily energy expenditure. How do you know how much it has peed? So you can estimate how much body water they have, and you're using one isotope to kind of set the baseline for the other isotope. So you want that relative change. It doesn't really matter the absolute amount that they've peed, but you have a baseline from the very beginning. So you know how much in the initial, yeah. So if you plot this for multiple species along that allometric relationship, this answers your initial question. We get a slope closer to 0.96 when we included about 17 species total. It's still 17 out of 370. There's a lot to go maybe, but I'm pretty confident about this relationship. And so that's a little bit more reasonable, but it's still really high, right? A slope of 1 is still much higher than it is for the rest of the birds. They seem to have a scaling relationship that's closer to that of lizards in terms of the slope than. Did I put it here? Is that okay? Can you hear me? So another thing we can look at is there was this. Okay, some of the physics papers are very hard for me to understand, the allometry ones, but there's a series of papers by this author named Douglas Glazier on the metabolic level boundaries hypothesis. And I found this very intuitive and nice and easy to understand. And so I wanted to test it as well. But he basically found that your scaling slope depends on what you're spending energy on. So if you're spending energy on very heat or water regulation kind of processes, your slope is 2/3 or 0.67. And if you're spending energy on very high power muscle activity like flight or something else I'll talk about later, torpor, then your slope is closer to 1. So we'll back off of that a little bit and first look at daily energy expenditure, which I got. Yeah. Scatter plot, yeah.
Right. So each of those circles is one individual.
So the small dots are individuals and the large dots are species averages.
And when you do a species average, how much scatter is there between individuals?
It can be very variable, which I'll talk about now in this next section, yeah. But you can remove the giant hummingbird from this and not remove it, and you still get a very similar relationship. So that I checked for that outlier because there's a huge gap. It's like 1.8 grams to 10 grams or 12 grams, and then suddenly there's a 19-gram hummingbird. So there's a huge gap here in body size, but it doesn't change the relationship. So I went to was there another? Yeah.
That's a long scale plot that it spans like 3
It's huge. It's huge. It is. Yeah, they're very variable. So I wanted to know how variable is daily energy expenditure in the first place. There were 2 field sites that I went to collect data on this using the doubly labeled water method again. One is Harshaw Creek, which is very elevationally diverse, vegetationally diverse, lots of little microclimates for my students and hummingbirds to take shelter in. And then the other is Sonoita Creek, which is much flatter, drier, hotter. So I expected temperature to be a big factor influencing how much energy they spent in these 2 sites, because they probably have to thermoregulate a lot differently in these 2 sites. So this is the data using the doubly labeled water method. Daily energy expenditure is on the y-axis and the different sites and conditions are on the x-axis. So Harshaw is on the left. Harshaw is on the left and Sonoita is on the right. And then there's pre-monsoon and early monsoon within the site. So we had 2 different seasons of data in the same site for 2 sites. So these are the data. Each dot here is an individual hummingbird. And the colored points are individuals that we got in 2 different seasons. So we know within a population what is changing. And we know even sometimes within an individual in 2 different seasons whether their daily energy expenditure is changing. So in some seasons, like the red dot stays the same in 2 different seasons. So they're not changing their energy. That individual is not changing how much energy it's spending in a day. But the other 2 increase hugely after the monsoons start.
Question.
Yeah.
Are they the same set of birds that you're following?
If they're black, then I only got 1 sample, like 1 paired sample from them.
Okay. No.
The colored ones are the same bird.
Continue.
Yeah.
Is there any other difference between these individuals? I mean, were they the same gender or something?
Most of them are male.
Yeah.
Oh, the colored ones? Yeah. I think all of these, there might be a couple of females, but the majority were male. And it was all within.
I'm curious whether there's a difference between the red, the green, and the purple one.
Not any difference that I could measure and tell. Yeah. I would think not. Yeah, they were all within a range of point something grams of each other. I can check that. Yeah, I don't remember offhand. That's a good question. But I would have made a mass corrected version of this and checked as well. I don't think they would have changed significantly. Yeah, so what also struck me in this graph is if you look at Sonoita Creek in the pre-monsoon and early monsoon, you notice there's no overlap. So that population of individuals was hugely increasing the amount of energy it spent in these 2 different seasons. So what is changing in the environment that's causing this huge change? And one way to do that is to look at what components of the energy budget are changing. So you're spending time sitting now, you are going to walk later, you're going to eat, you're going to do other stuff, and you're spending energy on all these activities, right? How do you partition your day in terms of time and energy? That's what I wanted to know for these hummingbirds. So we do that by measuring them with what's called a respirometry system, which we talked about in the morning in Nitin's talk. And it measures carbon dioxide and oxygen in the breath of the animal. And that's a good proxy again for metabolism or metabolic rate. And we can do that in some cases by not even catching the bird. We can put up this respirometry system and put a tube at a feeder, and the bird comes in and feeds, and it breathes out, and we collect the air from its breath and feed it into our respirometry system. So in the wild, we can measure how much energy a bird is spending hovering, for example. But we can also do this by putting the bird in a box, changing temperature, keeping it overnight, getting different components of its activity budget. So I did this with the same hummingbird population. I modeled how much energy it would take to do different types of activity budgets. So in the literature, by using binoculars and watching them, people found that they spend about 15% of their day hovering, 15% of their day flying, and 70% perching, which is what you see in this second activity budget model. But I also modeled some much lower energy, lower activity kind of models, and one really high one where they're spending 80% of their day hovering and flying and only 20% perching. And what I found is that many of these different activity scenarios could match to the gray box plots are what I showed you earlier, right? The doubly labeled water, total daily energy expenditure. So these should add up to the box plot. They're independent measures. And I found that multiple activity budgets could explain what was happening in these three sites, but in these three sites and situations, but only this high activity model could explain what was happening in Sonoita Creek. And these bars include variation in thermoregulation, in overnight energy expenditure, and all the other things. And those could not explain this huge jump. The only thing that could explain the big change was if they changed how much time they spent hovering, flying, and perching. And so
This is 95%.
the what is 95%?
The error bar, is that 95?
No, it's not in the colored one.
No, the box has
The
an
box
error
plot.
bar.
Yeah, I think it's 95%. Yeah. So
At that plot,
yeah,
what you're saying is that pre-monsoon, it's not explained. I mean, the energy expenditure works out, it's dominated by.
hovering and flying. Correct. Yeah. Yeah, exactly. So what was happening in the environment that could be correlated to this change? We found that it was the floral abundance and density. So there were lots of flowers, very densely packed and clumped in the pre-monsoon, and especially in Harshaw. But once the monsoon hit, there was actually a drop in floral density and abundance. And so we hypothesized that the birds had to spend a lot more time and energy finding and getting between flowers once the monsoon hit, and especially in Sonoita, than before the monsoon and in Harshaw. So around the same time, I was doing my PhD and I was going through an existential crisis, which happens every year. But it happened during my PhD in the sense I was wondering if I was working enough. And I felt very inadequate compared to my peers. So I collected data on my own activity budget. Every 15 minutes or whenever I unlocked my phone, I would say what I had been spending time on. And I did this for 4 years. And I basically found that I sleep, this has changed now. I sleep for 33% of my day, which was eight hours a day throughout my PhD, which is great. And I spent about 5% of my day, which is less than 1 hour on average every day, doing some sort of exercise or activity, usually salsa dancing. And then the hummingbirds, on the other hand, are spending between 22 and 84% of their day on hovering and flying, which means that they're spending 3 hours a day at minimum and 13 hours a day at max on hovering and flying. I'm spending less than an hour. They're spending 3 to 13 hours hovering and flying. Hovering is 10 times more expensive than sitting still. That's a lot of energy, right?
How does that relate to flying? The energy cost of hovering and flying, is it more energy expending to hover around
Hover is
than fly?
twice as expensive as flying, yeah, as forward flight. But flight can also be variable. You can be really efficient or you can be slower and more inefficient. But if you assume efficient flight, then hovering is about twice. So, yeah, I realized that they were very flexible in terms of their activity budgets, very flexible in their energy budgets, and capable of much more activity than I am. And this led to a whole bunch of other research questions. I was observing these things at the whole animal scale, right? I'm looking at an organism and the environment and seeing. And so I was measuring its energetic needs at an organismal level, but I started wondering what's underlying these energetic differences and what's allowing them to undergo such a huge shift in their metabolism every day. So another aspect of what they do is heterothermy. And this blows my mind. I've been studying them for 11 years and I still don't understand how it works. And I really want to know. So they're able to switch from being endotherms during the day to ectotherms at night. So they're going between like a human way of existing and a reptile way of existing every day sometimes. And I want to know how they exist. How is this possible? So at night, they're not getting nectar and it's dark and they would otherwise be using a lot of energy to stay alive, as we've seen, but they're able to drop into this state of torpor. And how many of you know about hibernation? Almost all of you. What animals do we usually think of as hibernating? Bears. Polar bears, I think, actually don't hibernate. I think someone said, but bears do. What? Hamsters, ground squirrels, very famously. So those are marathon hibernators, the ground squirrels. And a lot of animals can do a daily version of this. Hibernation is like a multi-day, multi-week version of torpor. Torpor is like the umbrella term. And what hummingbirds can do is daily torpor. So on the, I hope this plays. This is like the key video. Okay, good. On the left, there's a hummingbird. This is a black-chinned hummingbird, 3-gram bird. It's asleep at a high body temperature. We're looking at its eye temperature because that has the least feathers covering it. So we can see the skin temperature the best there. It's less insulated than the rest of the body. And on the right is the same bird later that night in torpor. So these are thermal videos, right? It shows you the temperature of the bird through the night. And earlier in the night, they're maintaining a high body temperature. Their internal body temperature is 41, 42 Celsius. And on the right, it's about 10 to 15 degrees Celsius, depending on the species. 10. In one hummingbird species, 3. 3 degrees Celsius. Imagine. What? Same. So they can't get colder than the outside air. They go down to... No, this is in the summer. But it's in the desert. So in the desert, sometimes it can get cold at night anyway. So in this case, it was like 10 to 12 Celsius at night. And so their body temperatures are dropping as ambient temperature drops down to some minimum for that species. So some species won't go down more than 15, even if ambient goes down below 15. They'll thermoregulate to maintain that minimum when they're in torpor. So humans are usually at 37 Celsius, right? And these hummingbirds are usually at 41. Birds tend to be warmer than mammals. But humans can only drop by 2 degrees Celsius before we're considered hypothermic. And that's bad. We want to be externally rewarmed, get out of that situation. You're in some Himalayas getting frostbite, like that's not good. But hummingbirds can go through this 38 degrees Celsius temperature drop down to 3 at the coldest and warm back up the next morning and everything's functioning normally. That's like the crazy part of this. Their hearts are beating fine, their brain is working fine, they remember that they're a hummingbird and they have to feed on nectar. I just don't understand how they can go through this huge change every day. Yeah.
My understanding of how this works is based on protein structure, that they, at different temperatures, they have different structures and the pathways so do hummingbirds have only a set of proteins that function over a wider range of temperatures, or?
Good question. I don't know. That's what I want to find out.
But also bird brains, you said they remember their hummingbirds after they come.
Presumably, I mean, they go and feed the next morning, yeah.
Okay. But have you done like some cognitive, like things they can remember or something? I mean,
I haven't personally done cognitive tests. So you're saying I would take a bird that didn't use torpor and test its memory or something, and then a bird that did it.
Yes, because
it's a good idea.
I would also be like the nerve cells in humans and birds have similar structures, right? Neurons, or are they very?
Yes. Okay. Yes. Yeah. Don't study neurons. Yeah.
Yeah, we can get back to this. Yeah, there's a student. What's the difference between torpor and sleep?
Torpor is not sleep. And in mammals that torpor has been studied in, the question is, what is the difference between torpor and sleep? Mammals that sleep seem to mammals that use torpors and hibernation especially seem to come out of hibernation and be sleep deprived. So it's not a recuperative state. Immune function is suppressed. The neurons seem to retract their dendritic spines from each other when they're in hibernation and they reconnect them when they come out. They don't seem to do all of the maintenance, growth, those kinds of functions usually when they're in hibernation. There's some weird examples where they like keep reproducing when they're in hibernation. Very strange. But on average, it's definitely not sleep. It's not like, I wish I could use torpor, Anusha. No, you don't. Like, that's not, it's not a fun state to be in as far as I can see. So they're spending about 2 to 30% of their normal metabolism when they're in torpor compared to sleep, right? Do they also sleep? So I think they sleep. So they avoid torpor, I think, as long as they can until they hit some fat threshold. And in that time, they're sleeping. And then they drop into torpor and they come out of torpor about an hour before sunrise all the time. Like, no matter which species where it is, they usually seem to come out an hour before sunrise. And I think they might sleep a little bit then as well. Do they have bihemispherical sleep? I really want to study this, but they're too small. I asked someone who studies unihemispheric sleep. So some birds can sleep with half their brains, right? You've heard about dolphins or migratory birds doing this. And I wanted to study this in hummingbirds, but the guy who studies sleep in birds was like, no, you can't. They're too small. You can't put recording EEGs on there. They just won't work. But maybe someday.
Okay.
What enzymes are at 41?
Birds in general stay at 41. That's the usual core body temperature.
The crazy part is 3 degrees centigrade, right? That's the crazier part. I would request we will keep on asking and prodding her questions, but let it continue and then we can again stop over after maybe 10, 20, 15 minutes.
So humans maintain a high body temperature even when ambient, the dash line is going down. Hummingbirds can do torpor, where their body temperature drops down to ambient. And I found that they can also do something in between. They can use shallow torpor, where they drop by only 6 to 10 degrees Celsius. And so they have a lot of control over their metabolism and their body temperature. And I didn't expect that because no one has found that birds can do both deep torpor and shallow torpor. I thought it was just like a switch that flips. They either go down or they stay up. But they can also do all kinds of weird things in between. And even on one night, I found a bird that was in shallow torpor for some of the night and in deep torpor for some of the night. So I think they're really responding to environmental cues and deciding how their energetic strategies. There's also some really strange torpor in other animals. There are bats that can use hot torpor. So in the middle of the day when it gets really hot, their body temperature rises, but somehow they're able to suppress their metabolism and save energy somehow on really hot days. I don't know how that works. Yeah, I know. It's the same face that Shubha has. So how we studied some of this was we go to the field, we use this respirometry system that I already told you about, measure the oxygen consumption, carbon dioxide exhalation, and then use a thermal camera to measure the bird's surface temperatures. You already saw the thermal video. And so we made a chamber like this that can do both at the same time. And I collected tissues from hummingbirds that were asleep, in torpor, or entering torpor, or coming out of torpor, and extracted RNA and did RNA sequencing to look at gene expression differences in these different metabolic states, because I want to know what's keeping them alive. Initially, I didn't start by wanting to know how they enter it. My mind goes to how are they still alive when they're functioning at 10% of their normal metabolism. But I think most biologists don't think like that. Most biologists are like, what's the trigger? What's the protein? What's the gene that's causing the entry? So I think we could answer both questions this way. But some interesting things that I noticed were when we euthanize the birds, we have to decapitate them to collect their brains. And their hearts kept beating for 10 minutes after they were decapitated if they were in torpor. But if they were not in torpor, if they were in sleep, their hearts stopped beating pretty quickly. So I think this tells me even at an anatomical, like visible level, they're hypoxia tolerant. They're able to tolerate very low oxygen levels, and their hearts are beating pretty independently of the rest of their bodies if their brain is missing, right? And then if I hadn't killed them, they would have woken up the next morning and gone back to feeding, and their hearts would have been fine without any tachycardia, bradycardia, arrhythmia, or whatever other disorders there could be of the heart. If you did this to us, our hearts would not work okay. So we studied this gene expression with 2 students from Cornell and a researcher who's now an assistant professor there. We found that more genes are downregulated than genes that are upregulated, which makes sense, in torpor compared to normothermy, which is sleep, high temperature sleep. We found one interesting gene that seems to have a lot of different functions, but it seems downregulated in torpor. So this is gene count on the y-axis, normothermy, which is sleep, high temperature sleep, transition into torpor, deep torpor. And across all tissue types, gut, heart, pectoral muscle, some more gut, liver, lungs, except for the lungs. It seems to be downregulated across all tissue types. And we're still working on this. This is very preliminary data. I have some summer students who have taken this and are running with it now. But the circadian system seems to be upregulated. Keeping track of time seems really important in animals that are using torpor, even though they're functioning at 10% of their normal metabolism. And this is crazy to me. But they know that it's 1 hour before sunrise, even though there's no light cue, they seem to warm up and come out of torpor an hour before sunrise. So they're keeping track of time, but I think they're also synchronizing all these different activities across organs by keeping their clock system running, which hibernating mammals don't do because they don't want to do this every 24 hours, right, over 8 months. And then there's a bunch of genes that I don't know how to interpret yet that are downregulated. There's a lot of post-translational protein modification and keratin metabolism, which I guess you don't need when you're in torpor.
Loss functions, the same way as it
do we know that? So I know that the clock gene itself is upregulated in torpor, and that happens in daily torpor in mammals. But Cry1, Cry2, the same thing happens, and PER2 and PER3, the opposite happens, which is not the same as in hibernating, as in daily torpid mammals.
But
the basic function, I think, is the same. They have the same rhythm through the day, yeah.
Organisms which are either well adapted to hot temperature or have to face cold temperature, that have a series of proteins that within the planet. Because the buffer is changed, for both cold and hot temperatures, targeted. Have you looked to see whether you see these dynamic transition changes in both classes?
The question was, there's animals that can tolerate extreme environments like extreme hot or extreme cold like tardigrades. I think it's not on.
Yeah. Specific set of proteins that are
reasonably well studied. Have you looked to see if those are changing in
heat shock, I didn't find as being
the cold one?
The cold ones, we haven't finished looking yet. Yeah. We have to do that next.
Anusha, just a quick thing.
Yeah.
I think if you want to look at clock, I think you have to monitor at multiple time points to make sure that it's difficult. Post-translational modification is interesting because they also have a huge impact on clock. So you might want to be seeing whether there is some crosstalk there.
Yeah, it seems like we're starting to do top-go analyses and more cluster kind of stuff. And regulating RNA splicing seems really important as a process. So yeah, the problem is the genome isn't as well annotated, obviously, as the mouse and the human. So I don't know how to get to all of the post-translational modifications as well. But in terms of the clock, so we tried to get all of our tissues between 12 a.m. and 2 a.m. So at least we controlled for time as much as we could, except for the ones that were coming out of torpor, which always happened an hour before sunrise. But yeah, obviously time points would be great. If I can find like a pigeon that uses it, then I have unlimited permits to collect them, then I would do something like a circadian series. So there's so many other pathways that we are really interested in. Metabolism pathways, cardiac function, mitochondrial function is an obvious one. Fat storage and metabolism. And we have this giant data set now that we need to analyze. So my students are starting to work on this. But there's lots of interesting things like with the brain, the mammal brain seems to, in mammals that use hibernation, the hippocampus seems to be the last thing that goes in and the first thing that comes out of torpor. The hypothalamus seems really important. It houses the central circadian clock. It's also responsible for temperature regulation. And then we were wondering if the cerebellum has some importance because it hasn't been described in mammals as far as I can find, but that's where breathing comes from. So how you're still keeping some of your basal functions up when you're using torpor. Their breathing is really interesting. When you're watching them with the thermal camera, they stop breathing for 10 seconds and then take 4 deep breaths and then stop breathing again. And so they have this weird sporadic breathing pattern when they're in torpor, which is really interesting. Yeah, so then while I was interviewing at TIFR Hyderabad, I spoke to Manish, who works there on Drosophila mitochondria, and his work is crazy, first of all. But I realized that they move and change and fuse and divide and things, and they're much more dynamic than the pills that we see in our biology textbooks in school. And so he suggested, why don't you look at mitochondrial changes in these different metabolic states? When they're in torpor versus sleep, how are their mitochondrial sizes and numbers changing? So we collected some tissues to do electron microscopy as well. And we haven't finished analyzing this data yet because the person had a hip replacement surgery and hasn't gotten to doing it. But we'll start looking at this as soon as we get that data. Okay, so more broadly, heterothermy is, I think maybe you're convinced that it's a very interesting strategy to deal with environmental change. And our instinct is to say it must happen in cold environments, but it actually happens in a range of conditions. It happens when animals are preparing for migration, they want to fatten up. And so they use torpor to store energy, even if the environmental conditions are not that cold. They can use it when food is unpredictable, when there's not much food available, so your energy intake is low, and so you reduce your energy expenditure to not be in a deficit. But we know very little about torpor, and there's 11,000 described bird species now. We know about torpor in less than 1% of all of them. So we're also trying to take a phylogenetic approach and look across the existing data sets and plot where those existing data are on a phylogenetic tree of birds. And so this is the phylogeny of all birds and the places where we have data on torpor. There's huge gaps. And I think we can use this to kind of direct our future studies to say, where else should we look for bird torpor, right? We can use this kind of comparative phylogenetics approach. And we can do the same in mammals. There's a ton of mammals that use torpor in hibernation, like an unbelievable number of mammals that use hibernation and torpor. So broadly, I have this idea of a comparative framework of heterothermy where I will study energy budgets. I could just not find any heterotherms in India. It's possible. I have some good ideas for where to look. But more broadly, I want to understand their energy budgets. And one component of that is to study their torpor use. So for example, we've started looking at animals on our own campus in TIFR Hyderabad. This is what it looks like right now. How many of you have been there? A few of you. So this is the hangar building where I sit. This is our main building, and we're going to have a lot of construction coming up here. And I want to study sunbirds and nightjars on this campus, which are both present. I'll tell you why. And basically integrate information about the environmental factors, the food availability, behavior, energetics, and the molecular mechanisms by using methods like this. We'll use weather stations, collect diet information, measure the activity budgets, like I've already shown you, the metabolic rates, and then in the longer term, do more molecular things. And I think these groups, just in my own backyard, offer a really interesting pair of comparisons, because in this phylogenetic tree of birds, the hummingbirds and a group called nightjars are very closely related. They're outgroups of each other, but they look completely different. Have you seen a nightjar? Most of you haven't, right? They're pretty large. They're insectivorous birds, and they are nocturnal. They're active at night, which is probably why you haven't seen them. You might have heard them. And they also drop their body temperature by more than 20 degrees Celsius. So there's only 3 groups of birds that can do that, that we know of. Hummingbirds, mousebirds, which are in Africa, and nightjars. And hummingbirds and nightjars are a nice pair to compare, right? So they're ecologically very divergent, evolutionarily very close. And on the other end of the spectrum, there's hummingbirds and sunbirds. So sunbirds are in this group of passerine birds or perching birds. There's 6,000 species of perching birds out of the 11,000 total. And they look very similar to each other, the sunbirds and the hummingbirds. These are the ones you've seen if you haven't left India and you think that you've seen a hummingbird, right? So they're small, they're iridescent like the hummingbirds. They have these beautiful, colorful feathers. They're nectar feeding just like the hummingbirds. They also eat insects. And they're separated evolutionarily by a lot of time. So they're evolutionarily very distant, ecologically very convergent. So I think there's a nice pair of species pairs to compare energetics in. And geographically, they're completely separated, right? The sunbirds are in Europe and, in Africa and in Asia, and the hummingbirds are in North and South America. So we did some preliminary sunbird point counts on our TIFR campus. And we found that there's some hotspots for where there are more sunbirds, which is really cool. My student Kavish, I forgot to put his photo on this slide, I'll show it at the end. But he estimated that there's about 96 sunbirds living on our campus, purple sunbirds living on our campus, at least in the summer. And then another student of mine, Rutu, over the summer, put out audio recorders all over our campus because these nightjars call at night and they're hard to see. So it's better to acoustically monitor them than to go and look for them. And she found that we have unfortunately, a very high abundance just where there's going to be a lot of construction. but also here in the back of the campus. And so she and my new JRF Swati have started making GIFs like this to look at abundance and how it changes across hours of night. So we can estimate when best to go out and trap them, when best to, we'll have an ecological set of data to then base our physiological studies on. So this can lead to a lot of interesting interpretations. We can test for evolutionary convergence and divergence in many ways. So a lot of these animals that hibernate and use torpor, at least colloquially, seem to have very long lifespans. We haven't tested this in a systematic way yet, but hummingbirds, you'd expect them to live 1 or 2 years for their body size, and they live 7 years on average. The longest a hummingbird has been recaptured at was 12 years old. So they can live unusually long lives, same as bats that use torpor in hibernation. We can look at the evolution of heterothermy, the evolution of endothermy, because birds and mammals came up pretty independently with the reptiles, non-avian reptiles in between, and the non-avian reptiles are ectotherms. So how did endothermy evolve in these two distinct groups? And how did heterothermy evolve in these two distinct groups? Did endothermy evolve via heterothermy? We can answer these questions by looking at this in this comparative framework, looking across birds and mammals and maybe even non-avian reptiles. There's also a lot of interesting biomedical applications. So people have found with hibernating mammals that they accumulate these tau protein plaques in their brains, and they can get rid of them when they come out of hibernation. And tau proteins are probably more of you know about this than I do. But in Alzheimer's patients, they accumulate these tau protein plaques and they can't get rid of them. We don't know why, whether it's a causation thing with Alzheimer's or it's just a correlation and it's not affecting them. But if we could figure that out, that would be useful. We can also look at cardiac function because they're able to slow down their hearts and still survive and maintain function when they come up, right? Animals like bats that are dying out in huge numbers in North America are affected by this fungus that affects them by affecting their breathing when they're hibernating. So their immune function is suppressed when they're hibernating, and this plays a huge role in disease and transmission. And then the clock genes are obviously a really interesting aspect of all of this. How do they keep track of time? And then there's all kinds of, we can safely cool and rewarm humans, then we can perform safer surgeries. We can extend the time that we have for surgeries. If we're able to do this for stroke patients or cardiac patients, we can buy a lot more time if we're able to cool them down safely and rewarm them. And then I read a lot of science fiction. There's sending humans to space, which I think space agencies are starting to fund symposia to think about this, right? If we can cool humans down, we'd save so much of the payload on spacecraft because we wouldn't have to give them food. We wouldn't have to give them as much air. We wouldn't have to clean their poop. Like it would just be a lot lighter, a spacecraft, if you could have humans hibernating. But would we be the same if we wake up? That's the question. So there's a lot of other interesting projects that I'm starting on. There is a hibernating animal in India that we know of, these marmots on the right, and they live in the same place in the Himalayas as pikas on the left. Pikas do not hibernate, and these places get as cold as minus 20, minus 30 degrees Celsius, minus 20 degrees Celsius. And so Nandini at IISER Tirupati and her PhD student who recently graduated, Harsha, who's very happily joined my lab, happily for me, joined my lab as a postdoc, have been studying these mammals in the Himalayas. And so we could get more physiological with them. They've been doing a lot of very interesting biogeographic and ecological work with these species. And then in the Northeast, in Arunachal Pradesh, Umesh at IISc has been studying birds there for 15 years, and he has incredible data on their survival across this elevation gradient and across different habitat types. And so, for example, he and Akshay, his student, found that birds that lived in logged forest, the same species, in a logged forest and a primary undisturbed forest, when the two were very similar in their environmental niche, the birds in the log forest did better. But when the log forest was very different from the primary forest, the birds in the log forest did worse. Does that make sense? But why? What is different in the birds in the log forest versus primary forest? So we can start to look at the physiological mechanisms that are underlying this change across habitat gradients, across rural versus urban, across logged versus primary, across low elevation, high elevation. And then there's some very serendipitous things that happen, I think, when you're at a place like TIFR Hyderabad. So Purnati is a recently graduated student, now postdoc of Tamal. And she has been attending my comparative physiology course, and she came and asked me if I have feathers. And so, yeah, I said I have some feathers of hummingbirds, and she wanted to image them. So she does confocal imaging. She also does some light microscope imaging, and she looked at my hummingbird feathers under the microscope. They have these iridescent parts. So you know what iridescence is, right? You look at the feather in different angles, it's different colors because of how the light is reflecting off of it. And that's from the structure of these little packets of melanin called melanosomes in the feathers. Based on how many there are and how thick, how deep they are and how they're arranged, you get different colors with iridescence. So she's looking in with this confocal microscope. There's these little air pockets you can see here. This part is very different in structure than the part that's iridescent at the top. And so she's going crazy imaging a bunch of feathers. There's these iridescent parts of the feather that look very different from the melanized, the monochrome or white part. There's the head feathers of this Anna's hummingbird that I was studying and the body feathers. And they have these weird tips where it's very strange. I'm running out of time, so I shouldn't do this. And then there's these corded, like rope-like parts of different feathers. Now I found like a bunch of other feathers, and she's going and looking for feathers, and my students are looking for feathers, and now there's like feathers everywhere. And I don't know where this project is going, but it's really interesting. So my core interest is heterothermy, but really I want to study all of this and kind of tie it together with energetics and see how animals respond to environmental change. Okay, that's it. I'm going to thank people. And thank a lot of people in the past who contributed to this work. And I will take questions. Okay.
This is exactly why we have this event in this institute. Thank you, Anusha. You connected so many people in this audience, and the brain activity and metabolism just peaked in the institute. So, so questions, yes. There's Anand. I'll get to it.
Sir, great talk, Anusha, as always. So curiosity question. So there's been like a long-standing set of things about hibernation and torpor that argue, like you said, that temperature has never really been a primary cue for it, but that ultimately all the variable cues tie down to an energy deficit of some sort, right? And so energy deficits will induce torpor or hibernation-like behavior. So do you know if there's anything about whether the corollary to that would be that even in something with a fast metabolic rate, if it were provided with unlimited energy and unlimited access to a source of energy, would it then be much less likely to drop into torpor? Say if it's a low-level, lowland hummingbird with unlimited access to nectar, if that were possible in some way.
You would think.
Yeah, but
I like way overfed them once in a while, and they just used it all up and then went into torpor anyway.
So they'll send themselves into an energy deficit no matter what they're given.
I don't think it's a deficit. I think they're very careful about it. So a student of mine went on to do a PhD on hummingbird torpor, and he studied the fat level at which they enter torpor, and they modulate it very carefully. So usually it's like 4% body fat, they enter torpor. So they use their crop's nectar store, then they switch to fat metabolism, 4% hit torpor. And then if it's colder at night, they enter torpor at a higher fat percent. So they enter at 6% so that they can budget for coming out in the end of the night because it's expensive to rewarm. And then when they're getting ready for migration, it's 30%. So they're using it to put on fat. And even their lean mass stays the same, but their fat mass increases and they use torpor anyway. So it really depends on a bunch of environmental conditions. And they can regulate when they enter it. So they enter it at very different times, but they exit it at the same time every night. Yeah. Yeah.
Amazing talk. And I'm not a biologist, so probably a very nice question. So I was very curious about the different levels of torpor. And I think one figure you showed where the same bird went to first a shallow torpor and then a deep torpor. So first of all, why don't they do something more continuous?
Something
more continuous, like slowly increasing the depth of the torpor depending on whatever the fat is. Why are there these transitions? And then what really determines whether going to shallow torpor or not? And because when they are in torpor, they are probably not using their brain or whatever. So something else is determining, okay, enough shallow, now you need deep torpor. So, and then
I
it's
think
going
shallow might be like a dim version of sleep and might not be it. In mammals, it seems to be a continuum between sleep and torpor, and I don't know if that's entirely the case in birds. Like some mice, when they're in some form of torpor, can still move around, which hummingbirds don't do. But why don't they go more? So they take about 20 minutes to drop into torpor. And usually when they've been studied, it seems like it's some switch that flips and then they drop with ambient. And it's kind of like a passive decrease of energy and of metabolism and body temperature. Why do they do the intermediate? I think maybe they're trying to modulate between the cost and the benefit. So in torpor, they can't respond to outside stimuli. So if I poke one by mistake, which I did once, they can't warm up. They can't respond to me until they warm all the way back up, which takes 20 to 30 minutes, right? So there is a cost. And there are individuals that don't use torpor all night. So I think they're trying to say, okay, maybe I can go some time without using deep torpor. Let me save a little bit of energy. And then there's other situations. We studied birds at nests. There's only the females, they take care of the young, and we put thermal cameras at the females at nests and found that they largely avoid using torpor, but there were some nights where they use shallow torpor. So they're kind of able to modulate, save energy for themselves, but still keep their chicks warm somewhat so they don't get too cold.
And another quick question.
Yeah.
So is it at all also depend on, like, let's say on that particular night, there was a sudden drop in temperature because it snowed or something?
Yeah, so the only 2 nights out of 108 nights that we study the females nesting, the only 2 nights when they use deep torpor were rainy nights. So I think there is an environmental relationship. Yeah. Sure.
I was struck by what you said about the breathing.
Yeah, the
Okay.
sporadicness of it.
So I may be wrong, but when a whale goes under, it takes a breath and goes under and then uses whatever oxygen is in that breath and stretches it out for a long time with super hemoglobin and so on, right?
Exactly.
Is it known what birds do? As in when they are in torpor, do they have their lungs full of air and they're making do with it because they're not metabolizing? Or do they not care? I mean, I'm just...
I don't know. I don't think we know.
But it should be find outable, right?
How would you do it?
Have they taken the inspiration or not before they stop? They inspire.
I mean, they do. And then nothing. So presumably there's
at the end of the last ha, is there an intake?
You're like listening. I'll check. I'll see if I can see it on the video. I'm not sure. It's a good question. Yeah.
Ultimately, this was all of this. Right? It's trying to allocate the resources.
Yeah.
I'm just wondering if you have seen it in mating and non-mating season. There are, because mating itself is a energy intensive process and the production is energy intensive. I'm just wondering, have you seen a pattern? Because that would also change the resource allocation in some sense, because there's a major cost. Second thing is, which is more molecular, is if you have to do such rapid changes in temperature, the uncoupling of the mitochondria has to change dramatically. I'm just wondering what is known.
Let's do that. There's nothing. Mating season. So we try to not keep females in the mating season in captivity, and because they would be away from the nest, it would be complete loss for the nest. So we only studied males, and the males don't take care of the young. They do have mating behaviors like displays and territoriality and things like that. But we haven't done a systematic seasonal study, especially one that includes females. It's a very ethically tricky thing. And many of the species, we don't even know when the mating season is in Ecuador. Yeah.
I was just curious. Their feeding habits are more like insects in the sense that they rely. When I say insects, I mean like bumblebee. I mean, are they
nectar
solely dependent on sugars?
Also insects, because they have to get protein from somewhere, especially in the breeding season, they'd feed a lot on insects. We're just starting to quantify how much. It's kind of hard to know how much of the diet is insect versus nectar, but most of the time, the base is nectar and insects are like a protein supplement, is our understanding.
They don't eat seeds and other stuff. No, I'm just trying to get a sense of whether, depending on what their source of calories is.
Yeah, so for the first 2 hours of night, the same student who did the fat things did some isotope labeling of their nectar and looked at their breath and when they metabolize different parts. And he found that they metabolize carbohydrates for the first 2 hours and then switch to fat metabolism for the rest of the night after that, regardless of whether they're in torpor or in sleep.
fat from in the reserve.
They're presumably converting the carbohydrates into fat and then using that later. Yeah.
Yeah. So the reason I asked the question about unihemispherical sleep is that when birds enter this state,
Yeah.
They're usually looking out for predators.
Right.
You'll see them actually go out into inside the water or go away into some place where they're less likely to be hunted, and then go into unihemispherical sleep.
Mm-hmm.
Okay. Now, of course, if the bird is falling into torpor,
Mm-hmm.
then it's not able to look for predators at all. So I was wondering whether these birds in the locations that you have studied them have a large number of predators at night.
It's really hard to measure predation. So at nests, we know that they get predated, like the young get predated, the
At
eggs
night.
get predated. At night. So one hypothesis is that if they're a heat-sensing snake, then they can't detect a bird in torpor. And so it actually helps them avoid predation by things like a heat-sensing snake. But owls and some, I don't know if bats eat them at night or something. It's really hard to measure because it's hard to find a hummingbird on a branch outside.
So, so the reason I asked this again about like, when you connect the unique if they're falling into torpor every night, then it's likely that they're not
Hemisphere sleep. Why?
because then they're not worried about
Yeah, but there's other ones that do avoid torpor all night. So I think if the larger hummingbirds tend to use torpor less and the smaller hummingbirds tend to use it more. And I don't know if that's a purely predation cost analysis thing or if it's purely energy stored in their body enough to fuel.
You mentioned something about the color at the throat being visible in different colors at different angles. This is the thing.
Say something more?
Yeah, you said you'll talk about it later. Skipped it.
Yeah, I just wanted to show the feathers. They have melanosomes, and so the colors that are not iridescent come from pigments which are from diet. So if you have oranges and yellows and reds, they usually come from carotenoid pigments in the diet. Whereas the iridescent colors come from these melanosomes, which are pockets of melanin that are deposited in layers. That's all I was going to say.
It's a pigment color.
It's like a photonic crystal kind of a thing, arrangement. Air gap and structures you have this light trapping that's taking place.
So my question is
I don't know what I mean.
structure.
I don't know if I had one picture of it. Purnati sent me. Let me try to find it. One second.
There's at least one case where a single gyroid is not in a point.
Okay.
There would be.
Yeah, one.
Is there another question?
Yes, there is one more question.
Go for it.
Yeah, excellent talk, Anusha.
Thank you.
As always. Yeah. Yeah, I wanted also during like when you were answering some of the questions, you mentioned that they are very precisely calculating the amount of resources they have, how much energy they can, dedicate to different components of whatever they are doing, their behavior. And again, given that there is a very narrow window of error, like because the energetic cost will be very could be deadly. I was just wondering, do we know anything with respect to what kind of mechanisms are there in their physiology, which are, because these mechanisms will be just too extreme, right? As compared to the mechanisms that we have to sense the amount of resources we have and how much we need to allocate.
So how do they know they've reached 4% fat? How do they know that they've eaten too much or too little? Yeah, because they maintain body mass throughout the day. So they're not overfeeding at any point. They ought to stay light in order to fly.
So are the mechanisms similar? Like, do we know anything about?
I don't know if we know. I know that their glucose transport is different. They have a lot more paracellular transport and a lot more tolerance of high glucose levels in the blood than we are. So they don't get diabetic even though they're feeding on nectar all day. But I don't know if we know anything more than that. We know about their sugar preference. We know about the proteins that underlie their sucrose versus maltose versus fructose preferences and things like that. But I don't think we know the sensing mechanism specifically, as far as I know.
Okay, well, I think we have to end here because,
I'll just show this.
okay, yeah, please.
Those are the melanosomes.
Right, right. So they don't seem to be very ordered, but that's okay. We can discuss
On the
this.
left.
Yes, on the left, right. Oh, that's
Those are the melanosomes, actual like EM photos of the melanosomes, and they're very layered and structured like that. Yeah.
okay. Fine. Thank you very much, Anusha. This was fascinating to say the least. And we're all thankful you're here and sharing your time with us. And we would tell the department that please send us more such colloquium speakers and so that the room is filled up. So thank you very much, Anusha.
Thank you all.
Let's walk together and have some snacks, okay? And we can ask more questions if there are.